Oh, good afternoon, everyone, and thank you so much for joining us for today's webinar, Seeds, Sods and Shoots, Restoring Tropical Seagrass Meadows. I'm Michael Rashid, and I'm joined today by members of the team, Tim Smith, Paul York, and who you can see on screen now. We had originally had Evie Furness, one of our PhD students, down to talk, but unfortunately she's unable to make it today, but we do have some of her work included in here. Um, yeah, look, thanks again for joining us. We're super excited to share with you our stories about seagrass restoration and the journey we've been on. But I wanted to start out with some acknowledgements. And first off, I'd like to acknowledge the traditional owners on whose land and sea country this work is based on. But probably deeper than just that acknowledgement, the work you see presented today is a true partnership with the traditional owner groups on whose sea country we are working, and you can see a whole heap of their logos on here. And Uncle James giving us the thumbs up as he's about to plant a seagrass sprig out in Meridian. So we're very thankful for those partnerships. Also wanted to acknowledge, as you can again see by all the logos there, we've got a lot of partners in this work, from our funding partners, industry partners, and the community groups, and other research institutions. And restoring seagrasses is really a collaborative effort. It leads a lot of hands on the ground, and we couldn't do this work without them. Um, quick bit of housekeeping as we go along. If you've got questions, if you can, it'd be great if you drop them in the chat online, and we can get to them at the end and answer them. But we'll also have an ability to, at the end, if you raise your hand in the Zoom meeting, that we'll be able to get to questions then as well. Just letting you know, the presentation is being recorded, but that won't include the question time. But it'll also be available on the TropWATER website. Apparently, there's a survey that pops up at the end, and we'd be grateful if you took the time to fill that in as well. So, why do we care about seagrasses? I'm just going to start out talking about the journey and why we're even bothering to restore them. And probably a lot of you online, I'm assuming, might not know an awful lot about seagrass, but there's quite a few species, around 72 species around the globe. They occur shallow coastal oceans around every continent apart from Antarctica. In our region in the tropical Indo-Pacific, there's 24 species, and in closer to home in the Northern Australia in the GBR, we have around 14 to 15 species. And that's important to consider when you're thinking about seagrass restoration. Not just one type of plant. You can see in the photos here quite a diversity of growth forms, life histories, and they require different conditions to grow. So, just like plants in your garden or your crops that you're planting in the field, you don't expect they all need the same things to get them to grow. It's the same with seagrasses. So we've got lots of seagrass. Queensland has, and Northern Australia has some of the largest areas of seagrass globally. But it's also important. It's not just lots of it. It does lots of really important things. So it supports lots of animals and habitat and food as well. And if you put some cameras out and do a day in the life of a seagrass meadow, this is some work from one of our team members, Abby Scott, from a few years back, with cameras out on the Green Island seagrass meadow. The place is alive with fish, alive with animals, and it has this productivity that drives a lot of that ecosystems that we have in tropical waters and coastal areas. We're also starting to understand, or have a pretty good understanding now. They're an incredible carbon sink. They suck carbon out through the atmosphere and through the water column, and bury it down in the sediments where they can lock it away. And they're really efficient carbon sinks. More efficient than land forests and in the tropics and the temperates and the places that often people are using for carbon sequestration methodology. And they do that because they stick it down in the ground and lock it in the sediments. They also bind the sediments together, as you can see in this photo. They have filters for nutrients out to the reef. They just do lots and lots of important things. So We've got lots. They're really important, but why do we need to understand how to restore them? Well, we're seeing more and more often in our part of the world, in the tropics, impacts associated with climate and weather patterns, and leading up to 2010, 2011, on Eastern Australia, we had these strong La Nina systems and lots of flood plumes going out to the GBR, multiple cyclones sort of capped off with Cyclone Yazi. And this created conditions that were really bad for seagrasses along the east coast of Australia, or east coast of Queensland, and and led to large seagrass declines across the board. Now, here's the image. This is from Cairns Harbour along the Esplanade there, a large seagrass meadow which disappeared around, or largely disappeared around the time that we're talking about over those 3 years. Since that time, the Cairns Meadow and other meadows in the region have had a general trend of recovery, but that wasn't the same everywhere. And in fact, even in this case, there's recovery, but it's taken more than a decade, and it's still not back to where it used to be. So, that's left a legacy of some places like Marillion, which Paul's going to talk about a little bit later on in more detail. But thriving seagrass meadows disappeared, haven't come back, and there's no propagules to support that. So, interventions required. And even in that Cairns example that I gave, there's 10 years or more we've lost all the services that those seagrass meadows were providing. So we really want to avoid that. And what we know from, climate change predictions and models, and what we're actually seeing right now, is that those kinds of La Nina conditions and those intense events are going to become more intense and perhaps more frequent. With seagrasses, we've still got time to act, because, while we have some areas that are lost, large areas of the seagrasses are still going quite well. So we've got time to actually understand how to restore these and how to intervene. And that's been a big focus of our group here at TropWATER and the Tropical Seagrass Restoration Group, is to actually get us into a position where we're ready for the future and we understand how to restore these tropical species. So for the rest of the webinar today, I'll move on to Tim's going to share with us the challenges of actually restoring tropical species and some of the solutions. Paul's then going to talk about what is one of the first meadow-scale restoration efforts in tropical Australia that's been underway, and some of the challenges and successes that we've had there. And then I'm going to talk a little bit about a slightly further afield location where we've been restoring a different tropical species in the Cocos-Keeling Islands, and a completely different set of challenges that are not of our making, that are to do with green turtles, and hopefully give you a flavour of where things are going and what we're looking at. So I will, with that, hand over to Tim.
Yeah. Where am I? Sorry. Okay, so today I'm going to talk about tropical seagrass restoration and some of the challenges that we face trying to restore seagrass in tropical habitats, particularly in North Queensland, and then what we're doing as a group to overcome some of those challenges, or how we're dealing with that, I guess. So just a quick. Yep, there we go. So tropical seagrass restoration in general, usually people use sods, so just a pot with sediment and the seagrass. You can use seeds, so you go out and collect seeds from the seagrass plants, then you go and plant them out later or soon, or you use fragments, which is just the rhizome of the plant, and you plant those out. So and we're starting to go quite well using these methods or variations on these methods. And it's becoming increasingly successful, particularly over the last 5 to 10 years, and particularly in some Australian context. So seeds for snapper in WA and South Australia. Up in Shark Bay, there's a lot of seagrass restoration going on now. There's Operation Posidonia in New South Wales, Western Port and Corner Inlet in Victoria, and there's programs kicking off in Tasmania as well. And so they're all starting to see some benefits and starting to see some restoration occur. And also internationally in Chesapeake Bay and in Northern Europe particularly, they're starting to have some really good successes with seagrass restoration. But if we think about in an Australian context, this infographic here is from a paper from 2020, 10 SL, and it was basically a review of all the seagrass restoration projects in Australia. And if you have a look at this map of Australia, you can see pretty much all of the restoration work has been done in temperate Australia, and on two or three temperate species, three temperate species. If we look at tropical areas, there's one lonely cross in Cairns and two species, so Thalassia and Somadosia. So there's just a bit of a mismatch there. There's a lot of seagrass in tropical Australia, yet we haven't really done any seagrass restoration. And there's also, as Mike mentioned, there's 14 species on the GBR, and so there's only been a couple of genera that have actually been looked at in tropical context. So this is where we sort of thought, all right, well, this is an area that we need to start doing some work in, as well as there's a need for it, as Mike just said. But with doing tropical seagrass or restoration in tropical habitats, there are quite a few challenges. Some of them are logistical. Some of them just relate to the seagrass and all the different species. And also the different habitats have different herbivores. And so in Northern Australia and the Indo-Pacific in general, there's turtles and there's dugongs that are mega grazers and they like to eat seagrass. So that can be a bit of a challenge as well. So at a logistical sense, trying to do seagrass restoration in a tropical context is difficult because particularly around northern Queensland and the Northern Territory, there's crocodiles and stingers in the estuaries and the coastal environment. And this is the main environment or one of the main environments where seagrass is found. So there's a real issue there because we can't get into the water. Also, a lot of these seagrasses are found in large intertidal muddy banks, and so it's really hard to walk on these banks. We can't get onto them, otherwise you'll sink knee deep into the mud, and they're often hundreds of meters long. So they're really hard environments to work in. And not only that, there's really low visibility water, turbid water, and it's often 50 centimeters deep, and you can't see the bottom. So being able to access the sites is a bit of an issue, and being able to see where you're going, what you're collecting. And this has really big implications for seagrass restoration, because if you can't access the site, it's really difficult to collect seagrass. How are you going to go out and get your seagrass if you can't get onto site? It's not in temperate environments. Generally, it's pretty sandy, or you can scuba dive down, you can walk out. We don't have those luxuries. So we've had to start thinking a little bit outside the box on ways that we can access our seagrass habitats for collection, then for planting as well. So how do we actually get onto the site to plant it where it needs restoration? And then lastly, also for monitoring. So this is a bit of a challenge as well, because how do you know if your seagrass is going all right that you've planted if the water's dirty the whole time? You can never see it. The low tide comes out every now and then. And how do we access it without disturbing it again? So there's like these range of challenges that we're working through and developing methods that we can do seagrass restoration. And so what we've started with was for seagrass collection. So the collection of the seagrass to then use for fragments and for sods is originally, or at the moment, we've got a pretty good process going where we rake up the seagrass. Or if we're doing sods, so if we've started using sods as well, and we use a post hole digger, and we just go over the side of the boat, push it down into the grass, bring it up and drop it into a pot. This can be challenging at times, even in itself, because sometimes you can't see the bottom. As you see in those pictures there, the water's pretty dirty. So we need to do a bit of background research and some mapping to find out exactly where the seagrass is prior to going out. Otherwise, you do a lot of raking and plugging of just sediment. So that's sort of in the collection. We also need to store our sods somewhere. And so it's hard that they take up a lot of space, and there's not enough room in the aquariums at here on campus. And if we did bring them in, they bring a lot of sediment, which isn't good for the other animals in the system. So Lloyd Shepherd, one of the staff members here, came up with this brilliant idea of a floating nursery, and basically got the square pontoons, and we made a float that stays in the docking cans, and we can hang baskets off there that are full of sods. And so this is really useful because we can collect them, leave them in there for a few days or a couple of weeks if need be before we plant them out. So it's just thinking outside the box a little bit on how do we store these? If we don't have the facilities, what are we going to do? So that's been a sort of just a lesson in trying to think a little bit differently. When we deploy our seagrass, so if we can't get into the water, how are we going to get it out? And so how are we going to put it on site? And so for Zostra, these Zostra projects we've got, and PY will talk about these, or Paul will talk about these a lot later, more in depth in the science and the background behind it all. But we're basically just tying them onto anchors, biodegradable anchors, and throwing them over the side. And we can still target certain areas because we've done our background work on the area, on the sediment and the topography, and areas where the seagrass needs to be, the old footprint of the meadow. And then we go up, we tie them up onto these anchors, and then we throw them over. We've got a project kicking off in a bit deeper water later in the year, and so we're going to trial some different anchoring methods then, using sandbags. Will these smaller anchors still work? But we've also refined this method a little bit, and one of our PhD student, Evie, who's unfortunately can't be here today, but she went to UWA earlier in the last year to use their big flume tank, and she used different sized nuts and bolts, or different sized anchors, and a few different materials to see when the seagrass and the anchor would start to move. So then we can really refine the best approach and the best anchoring method for the different habitats. So it's just sort of, I guess, some of the things we're looking at, some of the research we're doing to make our methods more efficient. And when we come to deploying the seagrass, so how are we going to do the sods? So we collected some sods, and then we're like, oh, okay, what do we do now? And so last year we got a hovercraft up, and we took it for a spin down at Marillion, and we were able to actually get on site onto the mud bank and plant those sods out. So it was a really good way of getting out to the whole mud bank, getting onto the site, getting into the right areas, and knowing that we're putting our sods and our fragments, we planted some fragments as well, in the right spots, and being able to access the whole mud bank, and being able to just get on there and move around, and get back safely. So that worked really well, except hovercrafts are pretty expensive, and they're not. There's not that many of them in Cairns. We can't just, or in Queensland or Australia, so you can't just get one next weekend if you need one. So we did a bit more thinking, and Lloyd again came up with a good idea of why don't we just use pontoons, floating pontoons. And so what happened is we're able to basically get the boat on site, wait for the tide to go out, blow up these pontoons. They're just little ones that go on the back of ski boats, use them quite often, and white boats. So just to float around in the water, and then we can access both sides. We can get onto the mud bank. We've actually found that we can not only deploy seagrass off those and deploy the sods, but we can collect it as well. So it's proving really, really useful. There are a few disadvantages. There's obviously that we have to sit on the bank for the whole rest of the day, but it's yeah, it looks like it's going to be a bit of a pretty good method for doing this. And so this is, I guess, making the point that it's a difficult place to work, but we're managing ways to overcome those difficulties, and we just got to, think a bit differently to what we'd normally do. And same with monitoring. So we've been using drones. One of the things that we've had a few issues with is basically last year there wasn't a daytime low tide that was low enough between November and March. So we're basically putting out a seagrass and we can't look at it for those long times. So that's another challenge. But we've got sort of some pretty good protocols now for the drones, and we're actually able to get a commercial drone pilot to run some mosaics for us in Cairns as well. So we could it's restricted airspace because of the airport. So being able to get that happening, so then we can monitor some of the seagrass fragments we put out there really closely now as well. So we're really moving forward in these kind of areas like monitoring. When we do our deeper restoration trials later in this year, again, it's going to be challenging. We're going to trial some underwater drones, some ROVs. Do we put cameras down? We've got to play around with that a little bit. So just really highlighting some of the challenges that we have in seagrass restoration in tropical habitats. The other one is herbivores, so turtles and dugongs. We've done a lot of work prior to our restoration work on looking at how much seagrass turtles and dugongs eat and like they're drivers of seagrass biomass. And Mike's going to talk about the implications of this for restoration later on when he jumps back on. The next sort of thing I was mentioning before, one of the challenges is that for the 12, 14 species we have in the GBR, there's not a lot of knowledge on their life history for most of them. So things like when do they flower, how many flowers and seeds do they produce, how do the seeds grow, when do they germinate, all those kind of questions, we're a fair way behind. So what kind of conditions do they grow best under? So we're trying to unlock a few of those questions for different species because that'll help us understand how we can restore seagrass better. Because if we don't know when they flower or when they set seed, it's really, really difficult to do seed-based restoration because we don't know when to collect the seeds. We don't know where the best places to go are. We don't know if we put the seeds out, are they even going to germinate or survive? So we've been answering a few of those questions through some different research projects. And so we've got some surveys and some work going on in Cairns Harbour with Zostra, which Paul will talk about a little bit later. Some surveys for Hologily and for Thalassia, looking at when they fruit. Some seed burial trials with Thalassia, which is another part of Evie's project. And some germination work for Hologily and Holophla. And this is just some of Evie's projects. So she buried Thalassia at different depths, different seeds at different depths. And just wanted to point out that it's just really informative for our restoration efforts because if the results show that at 1 centimetre depth, you have much better survival in the field than you did at deeper or on the surface. So that becomes really important in the future if we want to use seagrass seed restoration for Thalassia. We know that's the ideal depth to bury them to get the best returns. Similarly, we've been looking at growth dynamics, so growth under different scenarios with nutrients. Do apical meristems needed to grow quickly? And again, some of Evie's work with Hologily, she's shown that with Hologily, apical meristems apical meristems are the growing tips, so they're not necessarily really important for your fragment growth. So that means in practical terms that we don't need to choose fragments that have apical meristems. We don't have to be so picky. We can pretty much use whatever we can get. And we're developing this for some other species as well and sort of use shoot length and different things like that because it becomes important when you're trying to scale up how many shoots you can use. And then, so I guess we've started building a bit of a nursery. There's plans to develop one down at Marillion with Mandabara as well. Our seagrass, we've sort of got it growing really nicely in there now, and we're actually about to put on another 8 tanks. Will hopefully come online the next couple of weeks. And then, so we'll have some stock in those that we can hopefully keep on site and have those as sort of restoration stock that we could potentially get seeds from or fragments, or we could use in different ways. And then also that'll give us more scope to do some more of that restoration work, research into the different methods as well. And so I guess, yeah, just the sort of take home message is that we've had lots of challenges, but we're working through those, and we're working in ways that we can develop methods that are suitable for different habitats and for different species. And so I'll close up there, and I will pass over to PY, to Paul. Let me just jump in. Thanks everyone for listening. Oh, where's my Zoom gone? Stop sharing. Cool. All right, over to you, Paul. Thank you.
Okay. Um, Hopefully you can hear me. I just can't see the share button that's disappeared. Got it. Okay. Thanks for hanging around, everybody. I'm going to talk now about some of the experience that we've had developing and scaling up restoration methods for a particular tropical species around Cairns and Maroon Harbour, and give you some of the good and the bad experiences that we've had. Okay, so as Mike said in his introduction, back around 2009 to 2011, there was lots of seagrass loss up and down the Queensland coast, mainly due to La Nina weather systems that brought lots of rain, flooding, and tropical cyclones. And two of the areas that had quite large impacts were Cairns Harbour and Maroon Harbour in the wet tropics. So they're the two areas I'm going to talk about. Mike went through a little bit of the history there. Cairns almost lost all its seagrass but recovered, and took a decade to get back to almost where it was before, but there are still gaps there that we're doing experimental filling in of those gaps to see if we can increase or enhance the recovery. And Maroon Harbour in 2010, we lost the foundation species altogether there, and it hasn't come back, and we're looking at an intervention to bring it back through restoration. So the seagrass species we're talking about is eelgrass, or Zostra muelleri. It's a very widespread species in Australia, particularly on the east coast, all the way down to Tasmania. It's even found in New Zealand, and it goes up to Cape York and the Torres Strait. It's probably more of a temperate species, so we're up at the northern range of its range limits here in Cairns, and in fact, Cairns has got probably the last large seagrass meadows of this particular species on the east coast of Australia. Anything further here is just normally small isolated patches that you find. It's confined to estuaries and sheltered coasts, grows on intertidal mud and sand flats. And as Tim mentioned before, seagrass is a flowering plant. So this plant reproduces from flowers that are organised in spathes, which are like a modified leaf sheath. And you'll get 6 to 8 flowers in those sheaths. If they are fertilized, you'll get 6 to 8 seeds. The seeds have a semi-hard seed coat. The picture down here on the bottom right are the seeds. They're a little bit smaller than the grain of rice. And they can form a seed bank that will be dormant for at least a couple of years. So that's an area of recovery that this particular species relies on. So seed-based restoration is potentially a really good candidate for this species. And in fact, further south from here, Emma Jackson at Central Queensland Uni, and also Craig Sherman down at Deakin, they're using seed-based restoration techniques and working on researching them for this particular species further south. We know that there's been a seed bank here in Cairns because after those losses, we did a survey of the seed bank to see if there were any seeds that the seagrass could come back from. And the two maps here on the left show you the number of seeds and the number of viable seeds. The light yellow bubbles show you how many of the Zostra seeds were there in 2012 after the seagrass disappeared. And we've continued doing these seagrass surveys annually, and we're noticing that the number of seeds in the seed bank is continuing to decline. So we've also been, as Tim was saying, we've got to do surveys to find out when it's flowering, when's the best time to collect the flowers and the seeds if we're going to use them per restoration, and we've been going out and doing surveys for the last few years. We're finding a lot of flowers in the like immature flowers in the early reproductive periods, July to August. But then we're going out in successive weeks to see how they're progressing, and we're finding that they're disappearing. Any flowers that we do find are normally not in very good shape. They're starting to rot or get necrosis. And it doesn't appear that many of those are actually going through to maturity. And what we think potentially has happened is when the seagrass meadow contracted down to a few little patches, it lost a lot of its genotypic diversity, and there's a lot of inbreeding going on within the stock, and they're not actually able to produce many mature seeds. So with seed bank restoration out of the question up here in Cairns for Zostra, we decided to use vegetative sprigs, which Tim touched on before. That's probably the most common way that people restore seagrasses around the world. And for people who do a bit of gardening or tinker around their lawn, you're probably familiar with this type of thing. It's a little bit like taking a cutting if you're a gardener. So up in the top left-hand corner here is a sprig of Zostra that's been pulled out of the bottom. It's got an underground rhizome or underground stem called a rhizome with a growing tip at the end and a number of shoots growing up, and further along there'll be roots going down into the sediment. So we collect these and we started to do pilot studies to see how we could reproduce, how we could restore some of these meadows using these fragments, starting back in 2020. And we collected, we started off small scale because we didn't really know which method was going to be efficient and we'd be able to use, and we didn't have any funding. So we sort of started doing it off our own bat. So we just got 12 frames, which is a method of deploying the seagrass to the bottom, that bit like Tim was talking about before, weighing them down. This was a method that's been used successfully in North America. And we got 12 of these steel frames, tied 25 sprigs to each of them, threw them over the side of the boat at Maroubin Harbour, where we knew the seagrass used to live, and monitored them for the next year and then ongoing beyond that. 6 of the 12 still had viable seagrass growing on them at the end of the dry season. And then through the wet season, we couldn't really monitor because we didn't have good low tides. But after 7 months, there were only 2 of them that had, this is a timeline along the bottom of the slide. 2 of them had existing shoots that made it through the wet season. And then once they made it through that, they began to grow and spread. Yeah, it was sort of a bit of a bottleneck to get through that wet season. Once they did that, we had really good growth. The one down the bottom here was growing and expanding after the first dry season. It then made it through another couple of wet seasons and grew and expanded until it was about 3 meters wide, at times about 30 centimeters long and really lush. So it showed that we could grow it. So we went back the next year. So this method that we use with the frames, the idea is that once the seagrass gets established, you pick the frame up and leave the seagrass there. But it was a bit difficult because the water was a bit turbid. It was hard to find the frames, and it was soft mud that they were sinking into. So we decided people had been using biodegradable frames. We thought that might be a good way to deploy seagrass. So the next year we tried the biodegradable frames, which is this picture in the middle up the top here. It's sort of like a 3D mesh that's 3D printed using potato starch. And this particular model should break down within about 3 to 4 years in the environment, like compost down. And it's been used for restoration in mangroves, wetlands, seagrass, and a lot of different environments as a substrate to use. And generally in seagrass, it's buried just to the level of the surface. But because we could only drop them from the boat when the tide was in over the meadow, they weren't settling in all the way down into the mud, so they were sticking up a little bit. The 3D mesh was holding the leaves up, and when we got the really low tides in the middle of summer, the leaves were getting burnt off and they were desiccating and dying. So for this particular location, it wasn't a very good method to use. At the same time, we tried some individual shoots. Tim mentioned this a little bit in his talk, where we just tied individual anchors or weights, so that in this case little nuts, around individual shoots, and we put them in a group of say 20 or 30 and scatter them within about a meter and GPS mark those so we could go back and look at them and monitor them and see how they grew. And the timeline down the bottom here for the second year is following one of the 8 patches. Only one of them survived through the wet season, but after it made it through the 7-month period, you could see it was still hanging on. After they make it through that first wet period, they take off and grow really well. So we had some more success at a small scale with this method, and we thought this was probably the best method that we come up with. So we tried one more year. We tried using some different nuts and nails. Nails is the most common thing that's used in the Northern Hemisphere. Um, and we put 25 patches of seagrass out, seagrass patches through the meadow, and we had about 20% success rate that made it through the wet season. So we thought that that's actually a pretty good success rate for seagrass restoration, particularly using these methods. So we thought we had a method that we were ready to scale up and try and restore at a larger scale. So we applied for some funding, and we were really lucky to get 4 years of funding through BHP to help restore the meadow in Maruyan and also enhance the recovery of the meadow in Cairns Harbour. And we built on the local partnerships that we've been building through our pilot studies. We have partnerships with 4 traditional owner groups that are helping out with the restoration. 2 in Cairns, the Gimoy, Wallaburra, Yidinji, and the Yirrakanji ranges, and then Mandurah and Gundoy ranges down in Maruyan Harbour. We're also partnered with Ozfish Unlimited, who bring in recreational fishing volunteers to help, and we've got a lot of really good buy-in from the community in general. So we have volunteer days where people come and help process the shoots and help tie them up before we go out and spread them and plant them in the meadows. So that's working really well. It's been going for 3 years now. The first year we planted 8,000 shoots, and then we upped that to 16,000 shoots for the next 2 years. And along the way, we've been trying to refine the methods that we're using. As Tim mentioned, we've also tried pilot studies with sods starting last year, beginning with the hovercraft, mainly down in Maruyan, and then moving, as Tim mentioned, to the boat with the floating pontoons. And so if you look over on the right here, this is sort of some plots that were planted from July 2025. These were in Cairns Inlet, and you can see they were monitored again in October 2025, so 3 or 4 months later. And you can see they're actually starting to grow within that period. They then made it. These are the 2 same plots when they were last monitored in April 2026. So these two patches made it through the wet season and were continuing to grow. And now that they've made it through there, they're likely to take off and expand and start growing really vigorously through this next dry season. So reason for optimism there with this method as well. So that's some of the good news. The bad news is we've had some really big setbacks as well. And that's mainly to do with the weather that we've had over the last 3 years. We first started to upscale our large scale planting in 2023. And that was done sort of around August through to October. A couple of months after that, Tropical Cyclone Jasper came through, had a really big impact. It went a bit further north than when we are, but there was a couple of weeks where huge rainfall and flood events occurred, and yes, wasn't a great start to the life of the new seagrass shoots. The following year, we had again really heavy wet season. You can look at the map or the aerial photography here of Maruyan Harbour in December 2024. This is the next year, and you can look at that really dark, turbid water that's in there. Seagrass need a lot of light to survive, and when you've got these flood plumes that hang around, it makes it very difficult. And then again, for the 3rd year in a row, we were expecting to have potentially 1 or 2 of these years over the 4 and hoping we wouldn't get any, but we've had the first 3 years, we had even more rainfall last year. So if you look at the map of the rainfall in the middle here, the 3 years where we've tried to upscale our planting, we've had above average rainfall for all 3 years, and that sort of rainfall sort of mirrors the period that we had in 2009 to 2011, which, if you remember, that was a period where all the seagrass declined. So we've had really poor conditions for seagrass growth, and we haven't been able to get them established and survive through these 3 wet seasons. So now for the optimistic outlook. So it looks like the weather's about to turn. El Nino has just been proclaimed. I think late last week or early this week, they've declared we're going to have an El Nino, and that's going to bring with us a lot drier conditions, hopefully through the wet season, at least for this year and hopefully for the next few years. We know when we've got the good growing conditions that we can get these plants established. So we're very hopeful. We've got a lot more optimism this year of the forecast that we're going to have much more success. And we've also been very lucky to get our funding extended for a further 2 year through a grant through Queensland Government, Department of Environment, Tourism, Science and Innovation. So some really big hiccups to start with, but looking forward, we're very confident that we'll be able to get some positive runs on the board this year. So that's where I'm going to leave it and pass back to Mike once I stop sharing my screen. And we are going to we're going to start our volunteering days where we're going to start restoring in August this year. So the dates for that will be at the end of the slide. So if you want to come out and help us do some seagrass restoration in the next few months, those dates will be available. Over to you, Mike.
Thanks, Paul. And you couldn't ask for worse timing to have finally get the funding to scale it up. But looking forward to the next few years of getting that one up and running. All right, I'm going to round out the sort of case studies and that we're looking at by talking to you a bit about a restoration program that we've had growing Thalassia hemprichii, another really important tropical species. This time in the Cocos-Keeling Islands. And we're not dealing with weather, we're dealing with hungry green turtles, and it's a really interesting problem to have. For those of you who don't know, Cocos-Keeling Islands is an Australian Indian Ocean territory off the west coast, a long way off the west coast of Australia. It was declared an Australian Marine Park, or parts of it declared Australian Marine Park back in 2022. As part of that, there was a recognition that there used to be around 18 square kilometres of seagrass in the lagoon there that was pretty much lost some time ago. With the lagoon, it used to have seagrass over most of its footprint. When we went there to start this program, I mean, this is kind of the most seagrass we could find at any one site. It's was very little seagrass around, and I'm not going to go into the reasons behind that in too much detail. Suffice to say, it was probably a combination of a whole range of things that coincided at the same time. There was a small dredging program to establish a new landing facility, but also some very high thermal events in the lagoon, combined with lots of very hungry green turtles that have always been there, a really healthy population of green turtles. There's a lot of seagrass there to support them, but when things get out of balance, that due to circumstances probably not of their making in the original park, but there's certainly the major reason preventing current recovery in the lagoon. And we know from examples around the world that overgrazing from green turtles can have some pretty severe impacts to seagrasses. Where green turtle conservation is a real success story in lots of places around the globe, but there are sometimes the consequences for that, where the seagrass meadows, for whatever reason, may not be able to support the increased growth, and that's led to functional extinction of seagrasses from quite a few places. And we were quite concerned that was probably going to happen could happen in the Cocos-Keeling Islands. So when we first went in there, so we were confronted with that problem. We were also confronted with not a lot of information on how to grow Thalassia hemprichii, as Tim pointed out. It's one of those species. Evie's PhD has focused a lot on the seed-based side of that and some vegetative stuff, so we had some knowledge. But we had to go in there initially and trial to see whether we could actually find stock to grow it from, and then also a method that we could actually potentially get it to grow. We knew that the current herbivory pressure there back in 2022, 23 meant we had to cage out areas to trial it, but we also wanted to see whether those methods would work inside and outside the cages. And again, we were pretty much constrained to vegetative sprigs because you can imagine in that heavily grazed and low biomass area meadow, there was almost or very little sexual reproduction and not really any chance of collecting large amounts of seeds. We tried 3 different sprig-based methods, went out to a few locations and tested them out and planted inside and outside cages. And I'll cut to the chase because you guys have been very patient. We're cracking on for time, but after 12 months, this is seagrass planted in a cage, and you can see that all came from, or probably in total within there, about 15 sprigs that were planted across there. It's spreading and growing after a year, not much happening outside the cages. And we had a number of sites where that was an intertidal one. In subtidal locations, top row is photos inside the cage, bottom outside the cage, the same results. And when we looked at 18 months down the track, herbivory was the overwhelming pressure those blue bars are just the results over time for anywhere we had a cage, regardless of which type of restoration method we use. And but so that was one result. The other result too was we had really good success rates with whichever technique we chose, which showed we could actually get it to grow again, which was great. So at the end of the 18 months, when we were decommissioning the small cages, these photos were in the cage, we had really vibrant thalassia growing, and it's Chris holding up some fantastic thalassia, but also it started flowering and fruiting, and this was stuff that we planted, and that's pretty amazing in 12 to 18 months of restoration. Super great news. There we are. There's a nice little plot that we pulled the cage off. And here's 3 hours later, at the end of the experiment, some very hungry turtles getting stuck into it, which was no surprise to us. We kind of expected that was going to happen. And we'd actually already started the next phase of the planned approach to what we were doing, and that was actually scaling up the herbivore exclusion areas to larger nursery areas after a lot of consultation with the community. And we set up the first of these barriers, which are actually modified shark exclosure barriers that they use off the West Australian swimming beaches to keep people safe from white sharks. So they're not actually nets, they just stop the passage of large animals. Set the first one up in November 2024, and then once we had proof of concept, set up 3 more around the islands that have been running for some time. And we also used some of the material harvested from the first grow out experiment as propagules to start planting out in those barriers. And then very quickly, within 3 months, we started to see inside the barriers really good success, and outside the barriers not so much. And as time went by, but after less than a year, we were getting really good coverage inside the protection barriers, and there's a little bit of seagrass outside, very heavily grazed still. In January this year, we're getting close to the maximum density that you could expect in this type of seagrass meadow in the barrier. Outside the barrier, a few little struggling shoots still. And this was the last trip in April this year, the last sampling trip that we're on. And that intertidal site, you can really see we've got a highly functioning patch of thalassia that's full of fish and doing what you expect. You can see from that photo too that it's not doing so well when the turtles can still feed on it. So look, very interesting project, but it's not over. It's clearly not over. I mean, we can't cage the entirety of the Cocos Lagoon, but we've got successful restoration methods, and that currently those barrier areas are providing a really good in situ nursery, generating seagrass and seagrass propagules, and clearly things need to return to some kind of balance before you attempt to expand that restoration further outside. But that's certainly the goal. I didn't really talk about what's happening to the turtles. It was a very, very sad state of affairs. Lots of turtle mortality. As you can imagine, the major food source had largely disappeared. There's some evidence that things are, the herbivory pressure is reducing, but I think we've still got a little ways to go before we can start expanding this restoration effort outside of the protection from herbivory. But we're looking forward to it. So I just briefly now I'm going to switch to just touching on what's coming up for us. Had a little bit of an insight into some of the activities that we've got. We touched on some of the PhD work. We've got a lot of projects that have been looking at other associated things with seagrass restoration, like the return of services once you return seagrasses. And this is work from one of our master's students, Taylor, that just recently published. That looks at blue carbon in recovered and restored seagrass and the permanency of blue carbon stocks, which has some really big implications for valuation of the restoration success. Actually allows for cycles of fluctuation and the carbon stocks still preserved, which was pretty cool. We've got another PhD student. She's getting Darcy Philpott, who's getting very close to submission herself as well. She was looking at prawns and fish and the nursery function of seagrass meadows when they're lost and when they return, and how those communities respond to a restored seagrass meadow. Some really interesting things coming out of her latest paper, which has just been submitted on how that community might shift even though the seagrass is restored. Dushan, who's completed his PhD, or at least he's about to do his defence on Monday. He had a big project looking at modelling and connection between seagrasses up and down the GBR coast, and looking how that informs decisions about restoration and places you might want to source propagules, and also places that are isolated and need attention, which is really cool. I'm rushing through these, but there's some amazing work these students have been doing. And Evie, who was supposed to talk, but unfortunately had to leave the country for personal reasons for a little while. Her work is amazing, unlocking the code of particularly with Hologila uninervis and Thalassia impritchii, looking at all the different ways that you might go about restoring and ways to optimize that. And that work is getting some fantastic results out of that as well. So there's our students. What else have we got going on? As Paul mentioned, we've got 2 more years added on to the Marillion restoration, which is great. We're looking forward to that. We've got a new site coming soon. Don't look too carefully under the question mark. I wasn't supposed to see where it is. Some people might recognise that spot. But that's again another Zostermullerae study. Tim mentioned the nursery. It's under construction right now, a significant expansion, which is great news, and will free up a whole heap of stuff that we've got in the pipelines with other species. Really exciting. We're starting a larger scale restoration project for Hologila uninervis in Cowley-Curramine Beach. Thanks to the support from the Great Barrier Reef Foundation, and our traditional owner partners and Koch Foundation as well, supporting that, which is going to be very exciting. And we've got a whole heap of research on some of the other species we haven't mentioned for some of the other 14 that is on the way or just beginning. So we're really looking forward to that. Paul mentioned that we've got the next round of planting out in Cairns and Marillion, and there's some great opportunities for anyone out there, particularly if you're in the North Queensland area, to come and get involved. It's good fun for a few hours of preparing the fragments and getting them out there. And please contact Laura on that email or scan that QR code, which I'll leave up at the end of this talk, and if you're interested, and we can hook you up with a restoration experience. So look, I'd just like to say thanks to everyone for joining us, and it's good to be able to share some of what we're doing, and I think the exciting future of seagrass restoration exciting but challenging. It's a tough place to work, but it's nice to get some successes happening on the way.